Okay. Right, well, thank you first of all to Rob and the other organisers for the invitation. This is my first SVPCA, but hopefully not my last. Yeah, first. So I'm going to talk to you today about the avian tree of life, and in particular I'm interested in how climate change through geological time has shaped the diversity that we see today. So this is going to be very sort of technical and computational. So I'll start with a bit of background and my motivation. So this is a bit of a silly image. It hurts me more than it hurts you, but I do think it makes the point really well that we are currently experiencing a biodiversity crisis. We're losing species at absolutely unprecedented rates. So the jury is out on whether the current losses constitute a mass extinction or not, the sixth great mass extinction, but I think we're all in agreement that we are experiencing a crisis, we have a problem, and we need to take action before it's too late. So that's my motivation. So you might be wondering what any of this has to do with paleontology. So I usually give this talk or talks like it to audiences of ecologists. So I think that talking to paleontologists is perhaps going to be a bit like preaching to the converted. But regardless, I'll give you a bit of sort of background information. So I'm sure you're all familiar with the theory of uniformitarianism, this very famous sort of concept, Charles Lyell, the present is the key to the past. Well, I also think that the past is the key to the present. So we can look back through geological time, look at how climate change and how that's affected life on Earth. So the geological record contains all sorts of environmental data. So this includes things like global sea level change, atmospheric CO2 concentrations, temperature, and ocean acidification, and much more. It also contains, obviously, information on past life. So we can use this to reconstruct past climate and look at how that climate change has affected the shape of the tree of life. But if we want to know how the tree of life has been shaped, first we need to know what the tree of life looks like. So for that we need phylogenies. And if we're answering sort of macroevolutionary and macroecological questions, we really need big species level phylogenies, ideally containing thousands or even tens of thousands of species. So these phylogenies can include extinct taxa, extant taxa, or a combination of both. So, but a major challenge is still how we build these big phylogenies. So there's two main sort of methodologies, and in case anyone's sort of not familiar with these or needs a recap, I will run through the two methodologies now, so that it will be relevant later on in the talk. So we have on the left supermatrix approaches, and on the right supertrees. So very briefly, supermatrix approaches take primary character data, that could be molecular sequence data from extant species, it could be morphological data from extinct taxa. And we create a data matrix, and that's analysed to create a phylogeny. Supertrees, on the other hand, take existing phylogenies. So these can be from absolutely anything, molecular, morphological, any combination, and those phylogenies are turned into a matrix representation, and it's that matrix that's analysed to produce a phylogeny. So essentially, super matrices analyse primary character data, whereas supertrees analyse secondary character data. So I'll talk a bit about the advantages and disadvantages of each approach. So starting with super matrices, a big problem we have here is with missing data. So this is a data availability matrix. And you can see the big problem we have here is that for a very small number of species, we tend to have a large amount of data. And for a large number of species, we have a very small amount of character data. So that creates this massive blank area of missing data. We also have a problem combining different data types. So certain types of data, for example, for distance methods, that can't be combined into a supermatrix. So whether we should be using distance methods to build phylogenies is a different question, but if you do want to use it, you can't do it by supermatrix methods. It's also expensive. This is in several meanings of the word. So it's expensive financially. If you want to do molecular work, collecting new specimens and sequencing is really expensive. It's also very time consuming, and it's computationally expensive. It takes a lot of computational effort to analyse big matrices. And then, particularly if we're looking at fossil taxa, how do we reconcile different morphological characters, or even the same morphological characters, coded by different authors, published in different matrices? But of course, the big advantage is that they can tell us something new about phylogeny. That sounds really obvious, but it's not true for supertrees. So combining different data types, different genes, or molecular data with morphological data means that we can get this phenomenon called hidden signal. So we can find out new things about phylogeny interrelationships that the individual data partitions don't tell us. So supertrees. First problem is that only parsimony is tractable for large data sets. Now, there are Bayesian and maximum likelihood methods available now, but they're still limited to maybe 100 or so taxa. So if we're interested in big macroevolutionary analyses, we're still limited to using parsimony. So we have this issue of rogue taxa. So I should stress that this is not limited to supertree methods, but it is a particular problem well documented with parsimony supertree methods. So this is where, in the resulting phylogeny of a supertree, we find taxa that are placed in positions in the supertree that have absolutely no support in the underlying data. They're not biologically meaningful. It's really important that if we have rogue taxa, we identify them and remove them before undertaking any downstream analyses. It does seem to be a heuristic problem. Small data sets, maybe 200-300 species, don't seem to have this issue. But as data sets increase, we start to find these rogue taxa at a rate of 1 or 2%. And as I already mentioned, they don't tell us anything new about phylogeny. So if we do get so-called new relationships, we shouldn't be interpreting those biologically. A bit of a funny example, but I did bird supertree for my PhD, and the first time I ran this, I got an elephant bird nested nicely in the middle of the passerines, which is obviously nonsense. And it's a silly example because it's so obvious, but the problems can sometimes be a lot more subtle than that. So you really do need to know your group and look out for that. And another thing that negative of supertrees that I forgot to put on the slide is that you can't include taxa in supertrees if they haven't already been placed in phylogeny. So if you've got a new fossil of something, it needs to go in phylogeny before it can go in a supertree. But the positives are that they do provide an accurate, if we're careful, and quick consensus of the current state of knowledge. It is quick, it's efficient, and it's cheap. So there's the two main methods for making big phylogenies. So there's our big phylogeny made by whatever methods, but there's still something missing. So we can still use fossil record, geological history to inform our knowledge of this tree. And fairly obviously, it's fossils. So even if we don't have fossils in the tree, we need fossils to time calibrate the tree. So phylogenies, without fossil calibration, they only tell us about the relative divergences of the taxa. We need fossils if we want to know about when those divergences took place. So if we want to know about the shape of a tree of life, we don't just need the tree, but we do need fossils to see the shape of the tree. Right. So sorry for the slightly shouty large text, but this is a bit of a mishmash, but there's a lot of new results coming up, so I'm going to ask if people could just no social media or photographs from this point on, just to keep it simple. Thank you very much. Some acknowledgements first. There's some people here who have contributed in various ways to some parts of this work, and some institutions. Also a couple of funding bodies, BBSRC and the Leverhulme Trust, neither of whom specifically funded this research, but they have kept me in gainful employment and able to pay the bills so that I could do the research. So thank you anyway. So this is in two parts. So the first part is actually part of a collaboration that I'm working with Graham Lloyd, who's sat right there. And so these hypotheses may be slightly unfamiliar to Graham because it's so long since we first discussed this, so surprise. So we were working on the archaeology of life. This started around 18 months ago on a research visit to Macquarie in Sydney. So one of the nice things about being here today is that it gave me a really good excuse to come back to my vertebrate work after working on invertebrates for a few years. So we're interested in the archaeology of life, how the phylogeny has been shaped by environmental change through time, and also whether including and excluding fossil lineages changes that signal. So that's what I was going to talk about. It turns out this was completely overambitious and unrealistic. Oh no, there we go. So I'm talking about birds instead, which obviously are still a big portion of the archaeology of life. So I'll be talking about how environmental change has, or if it has, shaped the avian tree of life at the macroevolutionary scale. And I am afraid that the phylogeny I used here does only contain extant birds. So the next phase of this project is to do the analyses with fossil taxa included. So maybe I'll come back next year and tell you how I got on with that. So a bit of context here. So on the left here is, sorry, I'm sort of just randomly blinding the audience instead of pointing at the screen. So this is a tree published a few years ago. It's a really nice paper. It's a molecular tree. Bayesian has just 48 species, but it does represent all avian orders. And on the right is the main figure from that paper. So what they did with this tree, and Clement is one of my collaborators, so they use the TreePath software, which looks for shifts in net diversification rate through geological time. So the orange line is the net diversification rate through geological time, which is along the x-axis, and the blue line is global temperature through time. And the main result they found was that increases in net diversification rates in birds at this sort of macro level are associated with periods of global cooling. So it's a really nice result. It is only a very small tree, and they don't explicitly test for correlations between temperature and diversification. So that's sort of my starting point. So this is my methodology. So I took the Yeats tree of birds. So this was published in 2012 in Nature. So if you like birds, you're probably very familiar with this. It is a synthetic tree, but based on a molecular backbone. And it contains all extant species. Around 2/3 of those are positioned with molecular data, and the rest are placed taxonomically. So I took the 2/3 complete version of the tree, one's just based on molecular character data, and I ran it through the BAM software to model speciation rates through time. So this is an example here of the BAM output with time along the bottom, millions of years, and on the y-axis is speciation rate. So the thick red line is the mean speciation rate, and the purple fuzz is the variation. So there's 9,000 simulations there. I did 10,000 and removed 10% for burning, so that's why it's a funny number. Then I took 2 environmental time series. So on the figure here, just for visualization purposes, the blue squiggly line is global temperature through time. The little green line is a vegetation proxy. It's measuring C4 biomass. So increased C4 biomass represents the presence of more open, modern grassland habitats. Less C4 biomass is more forested, closed environments. So I did correlations for both of these time series against these speciation rates. And here are the results. So there's going to be lots of these histograms. You'll be sick of the sight of them, so I'll explain this one quite carefully. So along the x-axis is the correlation coefficient. This is temperature. And the y-axis is the number of times that I found that correlation. So it's a histogram because I did this for all 9,000 realizations of the speciation rate curve. So you can see that we've got most of the results are in the negative part of the correlation, whereas for those of you with good eyesight, there's a little bit creeping to positive. If we look at 95% confidence intervals, it's quite firmly within the negative. So that's suggesting that we've got high speciation rates with global cooling. So moving on to vegetation, this is a lot simpler, nice, even distribution. It's all in the positive confidence intervals. So we've got a nice positive correlation showing that we've got higher speciation rates with increased grassland habitats. So moving back to our hypothesis, environmental change does seem to shape the avian tree of life at the macro scale. So we've got high speciation rates correlated with global cooling and also with increased open grasslands, which could perhaps be as a result of forest fragmentation. So part 2, I wanted to look at whether we see the same pattern at the order level. So I used parrots. They're quite a small order, only 414 species, but I had the data for parrots. And also parrots are primarily, though not exclusively, forest dwelling. So I thought it seems reasonable that forest fragmentation might result in increased speciation, allopatric speciation in parrots. And second hypothesis, I wanted to see whether supertrees and supermatrices can both reliably reveal the same macroevolutionary and macroecological signal. So tomorrow Graham's going to tell you why supertrees are terrible, but hear me out. So this is the starting point. I've got 2 phylogenies for parrots, so there's 414 extant species if we include the recently extinct species that are extinct as a result of human actions. On the left is a supermatrix based on molecular data, 74% complete, and on the right we have a supertree, a bit less complete, 64%. So these are both fossil calibrated, but using different fossils, entirely different methods. And I just want to stress that these 2 trees are entirely independent of each other. So you can see these sort of coloured circles, don't worry about the details, but this is just the sort of the taxonomy. So it just illustrates that the trees are broadly similar, but not identical. The relationships are slightly different, all the branch lengths are different. So I carried out the same sets of correlations with paleotemperature and paleovegetation for speciation rates extracted via BAM for the parrot tree. And moving on to the results, more histograms. So we've got the molecular tree on the left and the supertree on the right again. So this is temperature. And we've got, so the results are slightly different. The shape of the histogram's a bit different. But if you look, I forgot to indicate the median, but if you look at the confidence intervals and the spread, it's almost identical. You'll also notice that there's no correlation, which is a bit disappointing. There's nothing going on there. So looking at vegetation, again, supermatrix on the left, supertree on the right. So again, we've got really similar distributions and a similar spread. So the median is edging a little bit into the positive here, but if we look at the confidence intervals, we can't, in all honesty, claim that there's any correlation here. So really disappointing result, but the data say what the data say. So we don't have any relationship between speciation rates in parrots with global temperature or global vegetation change. So there's obviously something else driving speciation in parrots. So for the second hypothesis, I wanted to look at whether supertrees and supermatrices can both reliably reveal the same macroevolutionary and macroecological signal. So I purposely set this up as whether they do tell us the same thing. I didn't want to use supertrees or supermatrices as the default because we don't really know which is the right answer. So the results I've shown you already suggest that they do give the same sorts of answers, but I wasn't convinced that sort of one example, sort of 2 trees from one group was particularly robust. So, and it's also not particularly realistic. So the trees are fairly complete, or if you work with vertebrates, that's maybe fairly normal, but I've worked with invertebrates for the last few years, and lots of people, myself included, carry out these kinds of analyses with only 10-20% of sampling. So, which is a more realistic scenario. So, I simulated highly incomplete phylogenies to see if the signal changes, obviously, or lack of signal. Could I create a false positive that tells us that maybe we shouldn't be using highly incomplete trees, but also do the trees do something different? So this is roughly what I did. So I started off with the two trees, the supertree and the molecular tree. So then I randomly removed taxa at 5% increments down to 80%, which left me with 50 or 60 species. And I mean, really, I shouldn't be doing these kinds of correlations on data sets that small, in all honesty. So anyway, so that's beyond realistic, hopefully. For each increment, I did 9 separate trees. I chose 9 purely because 9 histograms fits really nicely on an A4 piece of paper. For the publication, I'll be doing 100. And so then we have our subtrees. I don't know why there's 2 arrows there, pretend there's only 1. So we've got all these subtrees and then extracted speciation rates from them again for each subtree. So 9,000 again for each. And then carried out the same 2 sets of environmental correlations, resulting in lots of nice histograms. So that just took me about 20 seconds to explain that. There was an enormous amount of computational effort went into this. So this number is the number of individual analyses that I actually had to carry out, or the computer did, and I automated it as much as possible to actually get to the results I'm going to show you. And I'll be doing more for the publication. Right, so some results. So these aren't histograms, this is something different. So on the left, we have the temperature results, and on the right, we have vegetation. So the graphs are the same axes, and on the x-axis, we've got the percentage of taxa removed. So at the far right here, this represents the complete versions of the trees without any taxa removed. As we come along to the left, we remove progressively higher and higher proportions of taxa until we get to 80% here on the far left. So looking at the temperature graph first, so the dark blue is the supertree, the light blue is the molecular tree. So you can see that as we remove more and more taxa, as you might expect, they get more different. So this here, this distance, it's Euclidean distance, and it's a measure of the differences between 2 distributions. So as we remove more taxa, the distributions move further and further away from the original tree, as we might expect. But if you look at the supertree and the supermatrix, they actually pretty much track each other. And you also notice that the distributions for the supertree are entirely contained within the molecular tree distributions. So we can't actually tell these 2 apart. So whatever they're doing the same thing. So the vegetation, we've got some situation. They do, as we move more taxa, they do start to diverge a bit, but there's still this general trend towards bigger differences in the distributions, and we do have still a big overlap between the distributions. So they're still doing very similar things. So whatever they're doing the same thing as each other, but what do the histograms look like? So I'm not going to show you all the histograms because that would be painful, but just jumping straight to the 80% remove taxa. On the left, again, we've got the molecular tree, and on the right, we've got the supertree. So the yellow is the original tree, the whole tree that we started with, and the blue is the subtree. So this is the distributions of the correlations for the tree with 80% taxa removed. So you can see that for both distributions, they're remarkably similar. I did have 9 of each, this is just one random example. And more importantly, we have the same disappointing lack of correlation. But in this case, that's good. So no false positive. Moving on to vegetation, we find much the same thing. The supertree drifts a little bit towards a negative, but we still find overall absolutely no correlation between vegetation change and speciation rate. So it's a disappointing result, but it's good that they're at least consistent. So there you go. So second hypothesis, we do get the same result for both supertrees and supermatrices, and that remains congruent even when we remove up to 80% of taxa. And so whatever I throw at it, however much I try to destroy it, I can't create a false positive. So I'm cautiously optimistic that for this kind of analysis, we can rely on either supertrees or supermatrices. So very quick recap. So at the macroevolutionary scale, we do seem to have global cooling and forest fragmentation driving speciation in birds. But within parrots, we've got no response whatsoever to global temperature or global vegetation change. But supertrees and supermatrices are showing this congruent, stable recovery of the evolutionary signal, even at very partial trees. And I've already talked about this a little bit more. I will just say that I'm doing a similar thing with chondrichthyes. Very, very preliminary, but that seems to be showing the same result. And except that we do actually have environmental signal here. I don't want to say too much about it, but I'll give you a little hint. It's not vegetation. So thank you very much for listening.